So this is the second half of the how we metabolize fuel molecules video. And so we just finished the first part where we talked about how we break down amino acids, but this is the second page in the document that you have posted. And I really like this picture, so I want to go over this with you real quickly. So what you're seeing here, of course, is an amino acid as it's labeled. So this entire thing is an amino acid. You've got your carboxyl here, COOH. You have your amine here. The amine is NH2. There's the carbon, there's the hydrogen, that's your R group. This green thing here is the R group. So when we break down amino acids, of course, so remember, we've just eaten some protein. The protein gets hydrolyzed into amino acids. The amino acids are absorbed into the blood. And then when we want to break these down for energy, what we have to do initially is we have to break off that amine group, that NH2. I'm pointing to it right here with my marker. So the process of removing that amine group from the amino acid is called, and I'm zooming in on it here, deamination. You see right here with number 1 over it. Deamination is the removal of the amine group. So that leaves behind this part of the amino acid, which we call the organic acid component. It contains that carboxyl group that you see here along with that R group. Now, the amine group that we removed is toxic and we have to remove it from the blood. So it's going to get converted into urea. When it gets converted into urea, actually, this is the amine group that we removed. It gets converted here into ammonia first. So this is ammonia, NH3 is ammonia. That gets converted into urea. And then urea, which is our nitrogenous waste product, gets removed from the blood. So this is the urea again. Our nitrogenous, there's our N for nitrogenous waste. It gets put into urine. It becomes a big component in urine. And so there's how we get rid of it. So that becomes urine and it gets excreted by the kidney, gets carried within the urine to the urinary bladder where it's going to be then removed from the body. Okay, so the amine becomes ammonia, becomes urea, gets excreted by the kidney in urine. Right, there you go. So that is how we catabolize proteins for energy. Now lastly, you see lipids. So lipids, again, can be called triglycerides. Those are our neutral fats. They are going to get hydrolyzed into 2 monomers. They're going to get hydrolyzed into glycerol and fatty acids. So these again, this is a concept that we looked at in the biological molecules lab. So to continue, the glycerol will actually enter glycolysis. Glycerol has 3 carbons attached to it, so it's very similar to pyruvate. So it's going to enter glycolysis just as pyruvate would. The fatty acids, however, are going to undergo beta-oxidation. So that's a term you want to know. Beta-oxidation is a process that chops the fatty acids up into 2 carbon acetyl groups. These acetyl groups will enter this should be Krebs cycle, or citric acid cycle. If acetyl groups are generated faster than the cell can use them, they will be converted to ketones by the liver. Ketones are acids, and if they accumulate in the bloodstream, they will lead to ketoacidosis. Under conditions of starvation, the brain can use ketones to produce ATP. Okay, so we've talked about that idea that the brain either uses glucose or under conditions of starvation, ketones. So let me show you this fatty acid business here real quickly. So this is a fatty acid. A fatty acid has a carboxyl group on one end and a long carbon tail attached to that. And I'm just going to put dot, dot, dot rather than writing all of the carbon groups out. But these carbons are going to be covered in hydrogen. And this again, I'll review all of this in that PowerPoint that goes along with that biological molecule lab, so you can go back and listen to that again too if you have forgotten this information. But anyway, hydrogen's all already around the outside. So in beta oxidation, what happens is we come in and we cleave, we cut these, grab my different color, we cut the fatty acid tail, these carbons, into 2 carbon acetyl groups. So that would be a 2 carbon acetyl group. This would be a 2 carbon acetyl group. So we've got 2 carbons there. That's an acetyl group. Another 2 carbon acetyl group. And each one of these acetyl groups will then enter the Krebs cycle to produce energy and harvest hydrogen from. So, and again, I talk about the whole hydrogen harvesting thing in the videos that you're supposed to watch in regards to aerobic respiration, where I break down glycolysis, I break down the Krebs cycle. So, but these are the acetyl groups that will enter when we chop down chop this fatty acid into those 2 carbon acetyl groups. That process is called beta-oxidation, and it will generate excess ketone bodies in the bloodstream. In terms of what biological molecules gives us the greatest amount of energy production from its breakdown, it would be these. Fatty acids give us the greatest amount of energy when we catabolize them for energy. Okay, and that's it.